Okay, testing my mic. Okay, so my mic is okay, slide okay, camera okay. Okay, now it's already 8:01. So today we will finish up the chapter 10, chapter reproduction, development and growth. Okay, so yesterday we already covered all the topics about the sexual reproduction in human, and also the double fertilization topic about in the sexual reproduction in flowering plant. So today we will finish up all the remaining topics in this chapter. So yesterday I stopped at the double fertilization in flowering plant, right? So if you remember that after the double fertilization process, after the double fertilization process, you will form two substance, right? Two components, which are the zygote and also endosperm, right? So you still remember that there are two nucleus, right? So the generative nucleus will be divided into two sperm along the along the style, and then after that they will penetrate to the egg cell and also central polar nucleus. So when the sperm that fused with the egg cell will form the zygote, which is the diploid structure, and another one is will penetrate with the central nucleus, we call as polar nucleus, will form the endosperm, which is a triploid cell. So after that, after the double fertilization, similar to human, after double fertilization, human will undergo embryonic development, right? So similar to plant, they also will undergo embryonic development too. So for the plants that undergo embryonic development, they will produce the seed. So this topic is you need to know the formation of the seed through the embryonic development mechanism. Okay. So if you see this, sorry. Okay. So if you refer to this diagram, so this is the overall process of the embryonic development mechanism. You will see this diagram. This diagram will be the overall development of the seed in embryonic development. So if you refer that, the first step is the zygote structure. Zygote will be the first structure that form from the double fertilization, that when the sperm fuse to the zygote, fuse to the egg cell, will form the zygote. Why like that one? Okay. So zygote is already formed from the double fertilization process. Okay, after that you will see that the zygote will start to develop into the seed. So there are many stages you need to remember. The first step was that zygote will develop into two types of cells, which are terminal cell and also basal cell. And you need to think that you need to ingat, for only the terminal cell later will be developed into the seed. However, the basal cell is act as the anchor or support to the terminal cell only. So after development, the further development, you only will see that only the upper part is developed and change the structure only. But the lower part, you can see there are no structural change. Lower part is the basal cell, because it just only provide the support only. The main function of the basal cell is just provide the support to the terminal cell. Okay, after that you can see that the final product is seed lah, but inside the seed we have cotyledon. So this is the final product that will form in the seed embryonic development, which is the formation of the cotyledon. Okay, so we will see one by one. Okay, so the first stage is about the pro-embryo stage, meaning that it is a formation of pro-embryo. So you see this structure. So this is the ovule structure, and then already form the zygote. So zygote, I already mentioned, it will further develop into the basal cell and also terminal cell. So this one we call as pro-embryo stage. So after fertilization, your triploid nucleus will develop to form multicellular endosperm. So so this is the final the endosperm structure 3N, and then after that the endosperm actually is play the function to store the food in the seed. And then normally endosperm is used up during the development where the food is stored in the cotyledon. Okay, and then another one zygote, I already say it will undergo mitosis process, further develop into the embryo, and then further develop into the basal cell and also terminal cell. And I already mentioned that the basal cell will divide to supersensor cells. So we can see here. So they will divide to many supersensors, supersensor cells, which provide the anchor or support to your embryo. So the embryo actually is developed from the terminal cell. So the basal cell is only act as a support to terminal cell only. So terminal cell is the place for embryonic development rather than basal cell. So this one you need to know lah. And then supersensor cell will transfer nutrient from the parent plant or endosperm to embryo. Okay, and then also supersensor cell also will push the pro-embryo into the embryo sac cavity. And then the terminal cell later will divide by mitosis to become the pro-embryo, which attach to the supersensor cell. So this is the first stage, which we call as pro-embryo stage. So they will form pro-embryo lah. Okay. Okay, next stage is the globular stage. So why called as globular stage? You will see that at the terminal cell part, the upper part, the cell structure is in globular shape. The circular shape already. So that is why we call as globular stage of embryo. So normally this one is just the further development of the pro-embryo to become the globular embryo only. But the most important thing is the further stage. Once the globular embryo stage will form, later it will develop to become the cotyledon already. So either it can form one cotyledon or two cotyledon. So based on the plant types, if this plant is monocotyledon plant, meaning that it will only form one cotyledon. But if the plant is dicotyledon, meaning it will form two cotyledon. So that is why that monocotyledon names and dicotyledon names come from. Monocotyledon meaning it's only containing one cotyledon already only, and then dicotyledon meaning it containing two cotyledon. So this is the scientists, the plant biologists, classify the flowering plant based on these two classification, based on the number of cotyledon. So they found that some of the plant only contain one cotyledon, some of the plant containing two dicotyledon. So they directly using this naming as monocotyledon that containing one cotyledon and dicotyledon containing two cotyledons. So this is very important because in the exam you need to maybe got 1 year already come out the formation of embryonic development for the dicotyledon plants. Maybe coming out monocotyledon plants. So you need to know the number of cotyledons. So if you write wrong, because normally in your books, I think in Oxford they will mention about the it will directly mention about it will form two cotyledon. Then you just memorize it. But if the question asks about the formation of embryonic development for monocotyledon plant, and then you write it containing two dicotyledon, then you are wrong lah, wrong concept, and you are losing the marks lah. So you need to differentiate between them. And then normally once they already develop into cotyledon, will start starting to develop into the specialized tissue during this stage. So what is the specialized tissue? Like the ground tissue, vascular tissue that you already learned in semester 1, chapter 2. Right, ground tissue we had 3 types of ground tissue, right? Parenchyma, sclerenchyma, collenchyma, and then we also containing vascular tissue, xylem tissue, phloem tissue, all that lah. This all the specialized tissues will be developed from this stage after the development of the cotyledon. Okay, next I will proceed to the next stage. Okay. Okay, next stage it will continue to hard stage embryo. Why you call it a hard stage? Because it look like hard. It look like the hard stage, love shape. It look like the love shape already. So normally, just it is a further development of the globular stage. Just it will develop like a heart shape, love shape. So we call it heart stage. Okay. And then after that, it will further develop into the torpedo stage. It will further develop to the torpedo stage and will continue grow as the cotyledon elongate. Cotyledon elongate. So you will already see that actually cotyledon starting to develop already. Like this at the end of the embryo. Because I already mentioned after globular stage, it was starting to develop cotyledon. So when hard stage, cotyledon tissue already is emerged, and then further torpedo stage, the cotyledon was starting to elongate. And then as the embryo is elongate, it often curve back on itself and crush the supersensor cell. So it will curve. You will see that it will curve back on itself. You will see that it will curve back on itself. The cell will be roll backward, and then was starting to destroy or crush the supersensor cell. You can see that the supersensor cell now is already near the ovule wall already. And then the embryo cell between the cotyledon will differentiate into apical meristematic of the shoot. So you see that the embryo cell between the cotyledon, so normally at here something at here, you will see that stem tip at here between the cotyledon, right? So you can imagine that this two is already cotyledon structure, and then at the middle of this point, this is the place to differentiate into apical meristematic of the shoot. So that you can see that there is a stem stick So this is spatial for the apical meristematic of the shoot, and the embryo cell near the supersensor will develop into apical meristem of the root. So at here, root stem tip and root tip. At the end of the tips that near the supersensor cell is the formation of the apical meristematic of root tissue. Okay, after that, once they already go from the torpedo stage, after that it was starting to grow, become the mature embryo. So the seed now is already forming. It will already start to develop to form the seed. So you can see that the embryo structure is more larger, and the cotyledon now is already well developed, and then your stem apical meristematic and root apical meristematic cells are formed already, and also the seed cord now is already starting to develop. So this one is already final stage of the embryonic development of seed, and after further development, it will form totally as the seed now, like this structure. So you can see that the outer integument will form the testa, and inner integument will form the tegmen. So you see this structure. This is a final seed structure. You will see that the outer integument at here, the most outermost layer is we call as the testa, and then the inner part integument we call as tegmen. and then normally the central part of the ovule is broken down to use the embryo, and then the ovary wall will develop into pericarp, and then finally the seed tissue will undergo hydration and seed dormancy is induced. So it will finally to form the formation of the seed lah. Okay, so this is the overall process of the formation of the seed. So after that, we will continue to the formation of the fruit. So if you remember that after you form the seed, so later it will form the fruit, right? So you know that ovule is the development to form the seed, and then ovary is the development to form the fruit. Okay, so if you see this, ovary wall will be developed into pericarp. So this is the key structure to formation of the fruit. Okay, so the fruits are formed from the ovaries. The seeds are formed from the ovule. So this is the key important you need to know. And then fruit normally consists of 3 parts: epicarp, mesocarp, and also endocarp. So you see this structure. So you already form the seed at here, and then once the seed is well developed, it will form the fruits now. So pericarp is the initial structure of formation of the fruit. So after the pericarp is formed, they will develop to form the outer layer, which is called this 3 layer: epicarp, mesopart, and also endopart. So the fruit development will continue until the seeds are mature. So fruit development is more easy compared to the seed development. Okay, now we already finished up the sexual reproduction in flowering plant. Now we will proceed to the seed germination, the third topic of this chapter. Okay. So seed germination actually is the one of the continuous chapter from the chapter 9, plant hormone, about the function of the role of the gibberellin. So seed germination is the plant will use the gibberellin to initiate the seed germination. So you can see GA here. So this is the overall process of the seed germination. So once the seed is already produced in the plant, they will induce the seed dormancy first, because it containing the abscisic acid around the seed. The seed will start to germinate when they receive a lot of water. So if you see that the first step, it is the water. It requires water first. It requires large amount of water to wash down all the acetic acid and also break down the alluvial layer to release the gibberellin hormone to induce the seed germination. So we see this. So meaning germination is the process is for the seed to sprouting out. So meaning that this is the first initial growth for the plants to grow. Initial process to let the plant to grow. Okay. So germination is initiated by water imbibition first. Meaning that seed need to require large amount of water. You need to absorb large amount of water to expand the seed and also rupture the seed cord. To trigger the change of embryo to release the gibberellin plant hormone. So gibberellin will diffuse into the alluvial layer. So you can see here where the water is already coming into the seed. The seed is starting to absorb the water. The embryo was starting to release the gibberellin. And this gibberellin will diffuse into the alluvial layer to switch on gene to synthesize specific protein in the alluvial layer. So that the alluvial layer was starting to secrete the enzyme. So gibberellin is the plant hormone to activate the alluvial layer to secrete the alpha amylase, so that this alpha amylase will be released out from the alluvial layer cells to the endosperm. Now you can see there, these are all the endosperm. As I mentioned before, endosperm is the place that stored all the nutrient, organic nutrient, and organic nutrient and organic substance for the inside the seed. So especially the carbohydrate lah. So when the alpha amylase is released from the alluvial layer, it will go to the endosperm and hydrolyze all the polysaccharide, especially the maltase, especially the polysaccharide molecule lah. Not only alpha amylase, but alpha amylase is the key structure lah, key hormone, key enzyme that we produce from the alluvial layer. But besides from alpha amylase, it will produce the maltase and also protease 2, and diffuse into the endosperm to hydrolyze maltose, to form the glucose molecule. So glucose molecule. So this all organic nutrient will be transported into the embryo. So you can see that it will transported into the embryo. So now the embryo here, embryo to provide the nutrient to the embryo, and embryo was starting to grow now. So the germination is completed when the seed produce the vesicle and also plumule. So this nutrient will be used for respiration and growth of embryo. Because at the final step of the embryonic development of the seed, your embryo is already your embryo will stop to grow. It will form the seed. Will maintain at there only. Once the seed germination starting to occur, the embryo was starting to grow and to form the vesicle structure. So these are vesicle. So once the seed will produce the vesicle, so the seed germination is completed, and then it will further develop to form the plant. Okay. So this is the process of the seed germination. So it is very easy topic, and you should score it when asking in exam lah. So it already come out many times, I think 2 to 3 times in essay question already. So it is totally bonus mark to you when it come out. So hope everyone can master this topic lah. Okay, but apart from the seed germination process, you also need to learn about the factor affecting the seed germination. So there are many factors. One of the factor is water molecule. So as I mentioned before, if you want to starting the seed germination, you need to have the imbibition process by water molecule first. Your seed need to absorb water first. If the water molecule is not enough or too less, seed dormancy will be maintained, because abscisic acid is highly large amount in the seed, on the surface of the seed, to induce the seed dormancy. So you need to initiate seed germination, you need to remove all the abscisic acid first. So you need to absorb, seed need to absorb higher amount of water molecule. So water play the role to seed rupture the seed cord during the seed germination. So only the seed cord, the outermost layer of hardened layer of seed cord is ruptured, then the embryo can grow. And then not only water, oxygen. Oxygen is the key role for the seed germination too, because it is the initial development. So initial development of the plant growth. So the plant, the seeds, the cells of the seed is require high amount of oxygen. At this stage, the plant will not undergo photosynthesis yet. It will undergo cellular respiration first, like human, because it need to increase the metabolism rate to release energy for the growth of embryo. And not only oxygen, temperature also the key component to induce the seed germination. So in general, the seeds cannot grow when the temperature is very extremely changed, or maybe in very hot condition or very cold condition. So you can see that in many area, like the winter at the desert, for example, in the desert, you cannot see many plants. Why? Because the temperature at day is extremely changed. In the sunny day, the temperature is about 50 or 60 degrees Celsius, but at the night it is almost -10, -20. So it is very extremely changed, so that the seed cannot induce to germinate. So normally it is at optimum temperature lah, like human 25 to 30 degrees Celsius, like Malaysia lah. So that is why that Malaysia is the one of the place to ensure that one of the optimum place to let the plant to grow. Okay. And then also light intensity, high light intensity can increase the rate or during seed germination, and also the soil pH also will affect the nutrient and mineral need for plant growth and metabolism. Okay, so these are all the factors can maybe affect the seed germination. Okay, last we go to the last topic today, which is the growth topic. Okay, so this is the last topic today lah. So in growth, we will learn about the two main topic. Which the first one is type of growth curve, and next one is type of growth pattern. Growth curve and growth pattern are totally different things. You need to differentiate them. Growth curve and growth pattern are two different things. Growth curve is that you need to determine all the growth rate, the growth of every organism. Like human, you need to determine their growth curve, and fish or insect or giraffe, tiger, elephant, or you need to investigate bird. So all the organism can use to measure the growth curve. You need to see the growth rate. But another topic is we call as growth pattern. Growth pattern is only suitable, they classify the growth based on the different organism. Only this organism will follow this growth pattern only. Like human will follow the unlimited growth pattern only. Human will only follow the limited growth only. Human will follow the unlimited growth one. Limited growth meaning that we have the limited lifespan one. We will go until the certain age, then you will die. Unlike the unlimited is can survive along the lifespan anyway. Dia tak akan mati punya. So this is the growth pattern. Growth pattern only suitable for certain organism only. It is classified the growth based on the organism. But growth curve is the measurement to measure growth rate for every organism. So these two you need to differentiate them. You cannot too confuse between them. So in general, there are three types of main growth curve you need to learn: absolute growth curve, absolute growth rate curve, and one more relative growth rate curve. Okay, so normally this one you need to know that how to draw this curve. And then you need to know how to calculate the y-axis. But for absolute growth curve, you don't need to calculate because it is a parameter versus time. Parameter against time. Meaning that this is very directly, you just use any parameter. So normally in plant, like the parameter we use is either you use dry mass, or you use wet mass, or you use the other type of parameter. But in plant normally we use a dry mass lah. But in human maybe you use the weight, or you use the height, or you use the normally human is you use the weight or height lah, or you use the age like that. So this is a parameter against the time. So along the time, what are your parameter change? So normally in absolute growth rate, it will produce a sigmoid curve lah. Will produce a sigmoid curve one. Because along the time your growth will be increased. Because your parameter will be increased, right? And then after it will reach the maximum intensity, and then it will stop. So this is what we call as absolute growth curve. So this one you know calculation, you just use the any parameter. Y-axis is any parameter, and the x-axis is time. But when you see the rate, absolute growth rate curve, you need to calculate. You need to calculate, and maybe in exam, if want you to draw it, got 1 year it coming out in structural question. 1 year got coming out in structural question. Want you to calculate, want you to draw out how draw out the absolute growth rate curve. So this is a shape you need to have in absolute growth rate curve. it is a bell-shaped graph. So the y-axis is the change in parameter. Compared to first one is any parameter against time only. Maybe this one is after you calculate, maybe is 10 cm, 20 cm, 30 cm, 40 cm, 50 cm like that or 10 kilo, 2 kilo, 3 kilo, 4 kilo like that. But this one is change in parameter, meaning that this one you need to calculate. You need to calculate the difference of the parameter. By difference of the parameter. Okay. Next is relative growth rate curve. So this one is to show your efficiency of the growth, that is their growth relative to the size of the organism. Based on the size of the organism, you need to show that the efficiency of the growth. So the y-axis normally is you need to calculate the change in parameter, and also not only calculate the change in parameter, and also you need to calculate into the cumulative frequency lah. So this is a growth shape. So it will normally because it is a relative, so the first one will be 100%. After that it will decrease sampai it will along the edge. So when your curve is decrease very sharp, meaning that the efficiency of the lifespan of this plant of the organism is not very long lah, compared to when the curve is decreased more slightly. Okay, so these are the 3 type of growth curve. You only have 3 type only. And this all growth curve can be occupied any organism. Not only plant, human, and also animals can be applied. Use just only the change of the parameter only. Okay. Next, I will go to the growth pattern. Okay, so this one is very important, and you need to differentiate between the growth curve. Okay, so in growth pattern, there are many growth patterns, including unlimited. One of the is limited growth in human, and limited growth in human, and also unlimited growth in annual plant, unlimited growth in plant, and then allometric growth in human, and also isometric growth in fish, and also intermittent growth in arthropoda. So there are many growth patterns you need to learn. So we learn one by one. The first one is about the limited growth in humans. So I think this graph I don't need to explain very much. I think this graph you already learned from primary school until now. This is the basic growth pattern in human. You know that, in human. So it's a limited growth. So your growth will be increased until we reach the maximum, until it will decrease. When decrease, it will go to death phase. Okay. So the first phase is about the lag phase. So this is normally in early stage in baby lah. Baby for normally is 0 to 12 years old. Your growth rate is very slow. Very slow. And then meaning that at this point, your cell is cell division is very slow, and your organism during this stage is starting to prepare themselves, adapt to new environment. So this phase is the place to let the human adapt that kind of environment. So that's why some you can imagine why people can survive in desert, such horrible places. Because in desert the temperature is very high, extremely high. Why that people can survive at there? Like in Malaysia we already think that it's very high already. It's very hot. And then we go to desert, wah, terlampau tinggi juga. Or you go to the winter country. In Malaysia, if you go to winter country, normally many people will get sick from that in that country after you come back to Malaysia. Why? Because you are not adapt to that kind of environment. And why that people can survive at winter day? Because they already adapt to that environment during their early stage. Early age, they already adapt. So they already macam biasa je. Mereka macam biasa je. Tapi kalau mereka datang ke Malaysia, mereka tak biasa. Kerana Malaysia sangat panas. Mereka akan rasa Malaysia sangat panas. And the humidity in Malaysia is high. Compared to the America, the humidity at there is very low. But in Malaysia, is very hot and high humidity. They will feel sick at Malaysia after they come back to America because they already adapt that kind of environment in that places. Same of us, we already adapt to this environment in Malaysia already. Kami macam biasa je dengan Malaysia punya cuaca lah. Okay, so this is a lag phase. And another one is exponential phase. So this is the phase where your growth is increased very high. So normally this one is occurred when you are in your secondary school. You can find that when you enter secondary school, your ketinggian, your height and your weight will be increased very dramatically one. Maybe in 12 years old, in your standard 5, standard 6, you are the boy, especially the boy, more clearly to see. Normally the boy in standard 5 normally like 120 cm only. But suddenly it go to 17 years old, it came up to 170 cm. Suddenly increase 50 cm of the height. This is we call as exponential phase. Because your cell now is actively divide and elongate. So your growth rate will be now is very highest. Is the highest one. So that is why they're doing. If you in your secondary school, when you celebrate Hari Raya or Happy New Year, Happy Chinese New Year, and you get to your hometown, no long time, got 1 year no see, you can see that they will ask, they will tell you that you are more higher compared to last year. Last year see macam ni je, sekarang dah dia akan macam ni lah, will have the very extremely change at the secondary age. Okay, but when you enter after that you will enter stationary phase, meaning that at start your growth rate will starting to slow down and reach constant. Because now is your growth you're starting to reach maximum lah. Ketinggian akan tinggi lagi lah. Your height will not be increased anymore. Like you are 180 and then you normally is 180 already lah, or you are 170 and 170 cm, then you are only 170 cm lah. You will not increase your size already. And then normally this period is the growth rate will be limited by any factor. Normally is internal factor. Normally like organism your cells really reach your maturation, so we cannot divide anymore. And also external factor, but external factor normally is food. Your food or your environment also can reduce this. Next is maturation phase. This one is totally constant. You are totally constant and the growth is not increased anymore. So this one is normally is when you are high, more than 24 years old, you already graduate from your university. Then you are totally maintain your growth until you enter the elderly stage, until you go enter the census phase. Census phase is the aging process, meaning that your skin or your organ starting to decrease their functionality already, especially the skin. You will see that skin content will become more not more shiny, not more smoothly anymore. Go to awak boleh tengok lah, orang tua-tua tu, especially your father and mother, memang boleh nampak dia jadi tua lah. Kerana skin dia dah jadi tak macam orang muda lah. So normally when you are enter 60 years old lah, especially in human, when they enter menopause, they will starting to censor phase lah. And then finally everyone will go to death phase, meaning that this one is the place when the people will already starting to die lah. Some disease already coming out, like cancer or elderly disease, intermittent disease, or all these was starting to come out. Your growth rate was starting to negative, and your organs starting cannot function already, and then you was, and finally the people will be die lah anymore. Okay, so this one is human one. And then in animal plant, limited growth in animal plant actually same as the human, but the most different is at here. Before they enter the exponential phase, after the lag phase, the growth will decrease. You see here, the growth will decrease. Why? Because the initial decrease in weight is due to respiration or store food in the seed. You still remember that in seed germination, it requires the higher metabolic rate. So all the energy inside the in your food in the seed will be in high energy amount, so that it will decrease the growth. So you see here, this do the breakdown of stored nutrient in seed that later provide energy for the formation of the meristematic of shoot and root during seed germination. So this is one of the main differences between the plant. And another one is unlimited growth. So normally this one is only occur in secondary plant only. Secondary growth. Will not occur in human. Human is limited. We will totally die one. Until at the end we will die one. Unlike the plant can grow. It will grow unlimitedly. Okay, so unlimited growth refer to the non-stop growth over a relatively long lifespan. So it will grow unlimitedly. Dia kata mati punya, unless ada orang matikan dia, menyebabkan kematian dia. Unless got people destroy them, kill them. If not, they will grow unlimitedly. So unlimited growth consists of a series of smaller sigmoid curves. Actually, there are many sigmoid curves to produce like this. So this curve shows that there is an increase in mass and size every season. So normally however, although it is unlimited, so especially got the 4 season country, in autumn and winter season, your growth is slow compared to the summer and also the summer or spring lah, and summer and spring. Yeah. Okay. Another one is allometric growth. So this one is another type of growth pattern found in human. So this is the growth. The meaning of this growth is where the growth of the organ of an organism occur at a rate that is different from the rest of the body. Meaning that your organ growth is different from the rest of body. So more easy to see this. More easy way is your shape of the organism, the change in size of organism, which accompanied by a change in shape of organism. Okay, more easily you can see here this graph, meaning that the organ growth, the growth of the organ is totally different at all. Totally different at all. And then you see this at the initial stage, you can see that the head is very large. Kepala dia sangat tinggi punya, sangat besar punya. But at the end, your growth will be, head will be smaller lah, because head is the highest growth at the initial stage. So awak boleh tengok baby, kepala dia sangat besar punya. Kepala dia memang sangat besar, especially in the fetal stage. When you in your fetal stage, you go to, when during pregnancy, you can see the diagram by at ultrasound diagram. Awak boleh tengok kepala dia sangat besar punya. Why? Because head is have the highest growth at the initial part. When they're going a baby pun, after they are born, after the birth, you can see also the head of the baby is very big compared to us. Because height is the highest growth rate at initial, but the head after that it will stop lah. will stop at here, will stop. And then you will see that after that the lymphoid tissue will be starting to grow at the rapid stage when they already enter the enter the 6 years old, maybe 6 or 7 years old. The lymphoid tissue was starting to grow. And then reproductive organ is the organ that that grow at the lower the late stage of the human lah. It only was starting to rapid to growth when the human enter puberty age. So this is the pattern of allometric growth, meaning that the organ of an growth of the organ in the organism is different from the rest of the body. Okay, so it produces a change in size of organism, which is accompanied by a change in shape of organism. So this one, all this is the explanation that I mentioned according this graph lah. So you need to remember that kepala kami ni adalah organ, the most organ, organ yang the most the organ that will be growth at the initially, and lymphoid tissue will be growth at the highest when they already enter the first 5 year lah. Okay. And then another one is isometric growth in fish. This one is different from the human because the organ and the body growth are totally same. Compared to human, the growth of the human body, the size of the human body and the organ are totally different. When you see that this is a red line is the overall growth of the human body, but so you see that the organ totally different one. The head will be grow at the higher at the initial part, and then the reproductive organ will grow when they reach the puberty age. But in fish, it is very special. In fish, when the fish is grow, the size of the fish is grow, the organ will also enlarge, totally at the same time. Maksudnya apabila ikan tu besar, organ dia pun jadi besar, besar juga. So at the same time one. At the same rate and at the same time. So the size of the body or organism will change along the time. Now, meaning that this one is compared to human. Our human have the limited growth one. Dia akan ada satu limit punya. Memang ada satu limit, then dia akan stop. All the organ will stop. But compared to fish, size of the body will increase anymore. Because in human, our body size will be increased until a limited time, then you cannot grow anymore. But in fish, kerana ikan dia jadi besar and organ dia pun jadi besar, so dia akan jadi besar. So awak boleh nampak kenapa itu ikan boleh, if you not catch them, the fish can be grow at very larger size one. But this is the reason, because fish is according to isometric growth. So the change in size of the organism is, however, in this situation, change in size of organism is not accompanied by a change in shape of the organism. So the shape of the organism will not change. They only will change the size only, but the shape is not changed. So the relative proportion of the organ and whole body remain the same. Okay, next is about intermittent growth of arthropod organism. So one of the example is insect. But not only insect, all the arthropod like the prawn, crab, prawn, lobster, scorpion, insect, caterpillar, all these will undergo the growth of intermittent growth. So intermittent growth is like the ladder one. You can see it like a stair one. Macam tangga punya. The growth only will occur when the organism and the insect undergo ecdysis, and then the growth is immediately one. Why? So this is the discontinuous growth. Compared to the all these, this is the continuous. Karena dia increase the size continuously. Dia akan increase along the time one. Dia tak akan ada immediately and then suddenly ada growth and suddenly ada growth, tak ada. But intermittent growth is discontinuous, meaning that dia akan the growth occur, it will immediately occur and then will stop the growth again and then it will immediately again. So this is limit. So why the arthropod or insect cause this kind of growth? Because it is limited by the hard exoskeleton. So you will still remember that we have contained the type of the bone. We containing 3 type of bone, right? Endoskeleton, exoskeleton, and hydroskeleton, right? Endo, exo, and hydroskeleton. So if you still remember that in human we are endoskeleton, right? Endoskeleton, and insect and crab, prawn, scorpion, all that are exoskeleton. Exoskeleton. They are the hard shell. The hard shell cover their body one. So this kind of organism, because they are hard shell cover the body, so they cannot increase the size like human. Karena dia ada satu coverage at their body, so dia tak boleh increase the size simply because dia ada satu cover dekat sana. So only the place where growth can be occur, only the ecdysis or bonding process take place. Only the ecdysis process take place. So you see here, when the ecdysis process take place, the growth will start to increase immediately. So the hard skeleton does not grow but must shed for the organism to grow. So when the ecdysis process is starting to occur, they will remove the hard skeleton so the organism can start to grow. So the growth will occur very suddenly at rapid pace during ecdysis. So they will produce the intermittent growth pattern. So the stage between the ecdysis is known as instar. So yang ni, the growth in constant rate is instar. And then there were several stages of instar before the organism reached the adult stage. So in intermittent growth, the most important one is ecdysis process. So ecdysis actually is a mounting process which the old skeleton of the arthropod are replaced with a new soft layer and later becoming hardened layer again. So this is a process you need to remove the old exoskeleton first, and then starting to grow, expand their body, and then after that the exoskeleton, the hard shell will go back again. Okay, so this is the explanation about the ecdysis. Okay, so ecdysis is accompanied to metamorphosis. So metamorphosis is the change in shape occur very suddenly, and the process of the changing from one stage to the following stage in insects. So in insects, especially in butterfly, we know that the children of butterfly is caterpillar. Tapi tiba-tiba dia boleh jadi butterfly. Kenapa? Because of this process we call as metamorphosis. Because meaning that the stage, like in child stage, in children stage, in gamete stage, and they will go to children stage, and then after that they will become adult. The different of the stage will have the different structure, different shape anymore. The shape is totally changed. So this one is we call as metamorphosis. So there are two types of metamorphosis: complete or incomplete. So complete is totally you can see that the shape of the organism between in the different stage are totally different. So in adult stage, like the butterfly, adult stage is butterfly lah. But at the initial stage is A, and then after that it will become caterpillar, and then it will become pupa. After pupa become butterfly. So totally different. The shape is totally different at different stage. But for incomplete metamorphosis, the shape are totally same. Just the size is different only. But the change is occur very suddenly one. The size of the organism will also occur very suddenly. Okay. So for our syllabus, we will learn about the role of hormone in metamorphosis. So there are 3 hormones that you need to know. PTTH, the full name is protorexicotropic hormone. Protorexicotropic hormone, you need to memorize the full name. You cannot write short form PTTH only, especially in structural question and even in essay question. I already mentioned many times. In essay question, in the first sentence, you need to mention full name first, and then following you can use short form. So PTTH is the short form, and full name is protorexicotropic hormone. So this is the first hormone, and then another one is ecdysone, and third one is juvenile hormone. Okay, so you need to know what are their function. Okay, so PTTH, so the first one is your neurosecretory cell will secrete the PTTH hormone. So at here, so neurosecretory cell is in the brain of the insect. So it is like you will compare to human like the pituitary gland lah. But you cannot say in pituitary gland, but you will imagine in human like a pituitary gland. So in the brain of the insect containing neurosecretory cell, neurosecretory cell to stimulate to secretion of the PTTH. So this PTTH will be transferred to the corpus caudicum first and stored at there. So it will transport to the corpus caudicum and store at there first. And then this prothoracic gland will secrete the ecdysone when PTTH is released. So when your PTTH is released, it will stimulate the prothoracic gland to secrete the ecdysone. So what are the functions of ecdysone? It is responsible to trigger the ecdysis process and development of adult characteristic. And another one is corpuscle electum. So PTTH, when the corpus caudicum is stimulated, it will release PTTH to stimulate your prothoracic gland to secrete the ecdysone to trigger the ecdysis process. And another structure we call as corpus electum, or others book they say endocrine gland. So corpus electum will secrete the juvenile hormone. So juvenile hormone is the hormone that will inhibit the process of the metamorphosis. So juvenile hormone is inhibition of adult characteristic and retention of larva characteristic, meaning that it will juvenile hormone lah. Juvenile is like remaja, right? So they akan maintain the remaja one. Maintain their remaja. So in caterpillar, it will maintain the caterpillar stage only. So when ecdysone is secreted, it will trigger any ecdysis process. It will trigger ecdysis process at the growth of the organism, growth of the insect, and juvenile hormone will inhibit the adult characteristic. So you see this at the in the caterpillar stage, we call as larva stage, because of high amount of juvenile hormone. So every time ecdysis process, they will maintaining the larva stage, meaning it will maintain the caterpillar stage. This all caterpillar structure, caterpillar shape. But when the juvenile hormone decrease, metamorphosis will occur, meaning that the insect was starting to transform into pupa stage. Because the juvenile hormone starting to decrease, so ecdysone will take place to trigger the adult characteristic. So it will trigger the pupa stage first, and then finally juvenile hormone is totally declined as all, the zero concentration at all, and only have ecdysone. So it will trigger the pupa become the butterfly, to trigger the adult characteristic to become butterfly. So at here lah. So metamorphosis will replace the larva structure in the adult within the pupa, and when the JH is totally absent, the pupa will mount and become an adult. So like butterfly or moth lah, like that. So this is a total all about the role of hormone in metamorphosis. Okay, so I think that's all for today. So today we already cover all the topics about the reproduction, growth, development and growth. So next Tuesday I will continue the new chapter 11, homeostasis. So that's all for today, and that's all for today. So thank you for you guys watching, and see you guys next week. Bye.